Hello, I'm Tim Mitchison, professor of systems biology at Harvard Medical School, and this is a short recruiting video for the Systems Synthetic and Quantitative Biology PhD program. I actually just finished a short video as in my role as program director, so this is just a little snippet about the work my research group does, which might interest some of you. So I trained as a cell biologist. I've been doing research on how cells are physically organized my whole career. And these days, we're particularly interested in frog eggs. And you could say, why frog eggs? And I don't know how many of you have seen a frog egg in reality, but it's a single cell and it's really huge. We work on xenopus, so they're a little more than 1 millimeter in diameter. And when they divide, you can see the division planes beautifully just with your eye, even nicer with a dissecting scope. And they divide in the middle, and then they divide at right angles. And we want to understand how that geometry is established. So we know a lot of the molecules, but the question is, how do they arrange in space and time to take this absolutely huge cell, much larger than an ordinary cell, and divide it? And we use mainly microscopy for that. We have a lovely extract system we use. But so it's sort of fundamental science about how cells are organized. It's aiming to add to human knowledge without any immediate practical goal. The other side of my research group is much more practically minded. We're very interested in drugs and drug discovery. We're interested in cancer, inflammatory disease, to some extent, a neurodegenerative disease. I have a fascination with small molecule drugs, which some may consider old-fashioned, but they're the only kind of drugs that go off patent and become affordable eventually. So I think they're really very important. And it's kind of a miracle to me that you can take an incredibly complex system like the human body and ingest a chemical by mouth. And if things go well, and I know they often don't, you can significantly reduce the burden of a disease, treat the symptoms, sometimes even treat the cause. And to me, the human body is a sort of ultimate systems problem, a kind of black box. You're adding a drug on the input, and then you're looking for modification of disease at the output. That to understand that, we have to understand the molecular level, the cellular level, and then the physiological level. And in my group, we try to connect all three. Just for example, we've been studying drugs that perturb microtubules, which are also involved in cell organization. And there's a very famous ancient medicine called colchicine. It's been used since ancient times. It comes from plants and is used to treat inflammatory disease. And it was very unclear how depolymerizing microtubules would have a useful, beneficial effect if you've got inflammatory symptoms. And the answer turns out to be quite complicated, actually. The drug is taken up preferentially by our liver, which is a specialist in detoxifying drugs. And then it acts in the liver to trigger gene expression, and the liver secretes proteins that then move all around our body, we believe, and calm down white blood cells, which is doing the actual job of the drug. And that's, we just had a paper accepted on that, like at the beginning of 2021. So that's new knowledge. And now the question is, okay, given that knowledge, what can we do with it? Can we come up with better treatments of, for example, serious inflammatory disease like lung infiltration can easily kill you. It's the main thing in some sense that kills people with COVID-19. It's the immune system becoming overactive in the lung, and there's a big need for new treatments, and perhaps particularly affordable treatments that could be used worldwide. So we have some ideas based on our colchicine work. So it's not particularly mathematical in my lab. We do use modeling. We do a lot of image analysis, but we tend to be sort of interested in the type of problem, and then whatever methods necessary, and particularly microscopy. So modern optical microscopy is at the roots of all our projects. So anyway, that's just one snippet of a professor in the SSQB program. You'll hear from lots of others if you stay tuned. Okay, thank you. Bye.